directionality to the strands, and we have DNA and RNA as deoxyribonucleic acids and ribonucleic acids. Okay, and I say what is this 5 prime and what is the 3 prime, what is 5 prime to 3 prime directionality in the nucleic acid. Okay, so I say DNA strands always runs in the direction of 5 prime to 3 prime, 5 prime to 3 prime. Okay, what is 5 prime means, which is 5 dash or 3 dash, not very appropriate to say, but I'll go again back to the carbohydrates lecture and I'll start naming the numbers of carbon and like this. Okay, so let's see how we name them. Anyone still remember? 1, 2, 3, 4, 5. This 5 prime has a phosphate group. Okay, fifth position we have a phosphate group. Okay, and that's why they are named as 5 monophosphate or just 5 prime. Okay, let's go ahead and see at the 3 prime. Okay, you have a phosphate group here too. Okay, and this is 3 prime phosphate, this is 5 prime phosphate, and this is 3 prime phosphate. There was no phosphate here before. Okay, if you remember, there were no phosphate before. This phosphate comes from this group. This is an individual nucleotide, this is an individual nucleotide. You see that two nucleotides are joined together by phosphodiester linkage. I said it before. If you want, I can dive back to understand how the phosphodiester linkage has been formed. Okay, I'll try to make it here. So do you remember, if this is your sugar, there should be a OH group here, right? And this is your nitrogenous base, and this is your CH2OP, which is phosphate. Okay, do you remember, if you make a similar nucleotide here, you have a nitrogenous base here, it could be purine or pyrimidine, and you have a OH group here, and you have a CH2OP. Okay, and biphosphate means this should have a hydrogen, means P double bond O, this is O negative, this is OH, and this is OH. Okay, or if you write this H, it could be H3PO4. Okay, phosphoric acid. Okay, so remember, iPad is already suggesting me to make a bond from here to here, which is correct. Okay, so if you see, you have a phosphate here, which is OH and OH, you can remove a molecule of water. Okay, can you see that this phosphate could be attached from this sugar again? See, this OH and H can be removed as H2O, and this oxygen will have a bond with phosphorus, and this oxygen will bond with sugar. So that's what you see. You have a sugar here, you have oxygen here, you have a phosphorus here, you have a oxygen here, you have a CH2 here, and this is how phosphodiester linkage is formed. Did you get this? Yes or no? This is your nucleotide. If I see one more time, this is your nucleotide with a nitrogenous base, a sugar, and a phosphate, right? This is your nucleotide. Okay, you have another nucleotide here, but this one has a phosphorus attached with oxygen and hydrogen, and oxygen and OH will be removed as H2O, and this oxygen will bind with phosphorus oxygen. So this is OPO bond. Okay, and these two will again bind with H2, CH2 and CH2. This is why they are called as phospho, means there is a phosphorus, and this is ester bond, O and O, and this is why they are called phosphodiester bond. Okay, Clear? So this is phosphodiester bond, they are linked, and this runs from 5 prime, and at the end there is no nucleotide to be attached, and that's why they are called as 3 prime, and this is called 5 prime. This prime means free, 3 free phosphate. Okay, at the third prime you have free phosphate group, and at free prime you have free phosphate groups, but that's why they are called as 5 prime to 3 prime. Okay, if you see RNA, what's the difference between DNA and RNA? You have oxy group here, right, and there is devoid of any oxy group, so this is deoxyribose and this is ribose sugar. Similarly, 5 prime, 5 prime free phosphate, 3 prime free phosphate, and this is called 5 prime to 3 prime phosphodiester bond. Okay, you have a free, not phosphate here, but a free hydrogen. Sorry, this is free hydrogen, and this is free phosphate. Okay, there's no free phosphate here at the 3 prime end. Okay, so 5 prime means there's a free phosphate at fifth position, and at 3 prime means 3 hydrogen at uh, 3 prime position. Okay, and that's it. It always runs from 5 prime direction to 3 prime direction. So if I write something like this, like draw a straight line. Okay, if I simply draw a straight line with an arrowhead, you have to understand this is already 5 prime and this is already 3 prime, because it always runs from 5 prime to 3 prime, and this 5 prime means they have a free phosphate here and a free hydrogen over here, hydrogen or hydroxyl group. I would say hydroxyl group rather than hydrogen. Okay, this will have a hydroxyl group rather than hydrogen. Okay, but anyway, so if I run another strand like this one would be 5 prime and this one would be 3 prime, and that's why strands of DNA are anti-parallel. Okay, why strands of DNA are anti-parallel we will understand in 5 minutes. Okay, why they are not parallel to each other, like 5 prime to 3 prime and 5 prime to 3 prime. This is wrong and this is correct, and why we will definitely learn in 5 minutes. Okay, but I think I'm clear up to this point. Yes, let's move ahead and see hydrolysis of DNA. A few things to remember: RNA is unstable in alkaline medium. Yes, hydrolysis are driven by enzymes. RNAs are RNA digesting enzymes. Okay, similarly D and similarly DNAs are DNases are DNA. Yes. Okay, and similarly proteases are, yes, similarly lipases are, yeah, lipid or fat digesting enzymes. So ases means enzymes for sure. Okay, anything with ase are enzymes. Okay, whatever they digest they are named according to that. So is this a enzyme? Yes, this is an enzyme. Yes, so anything with the ase is enzyme. So can you tell me what is the difference between R and A and R and Ase? One is ribonucleic acid, other is enzyme that digest ribonucleic acids. Okay, cool. So dicer are another component. They are like, they are RNA but they are double stranded. So mostly what we know is RNA is mostly single stranded, double stranded is DNA, single stranded is RNA. But if I ask you what is a double stranded RNA, you will give example as dicers. Okay, they are mostly present in small group of bacteria present in leguminous plants, and they help us to eradicate infections. Okay, this is a very famous technology we know as RNA interference. Okay, They make antiviral proteins and they protect from viral infections. Okay, mostly they are present in plants, which are responsible for protecting genomes by slicing the RNA as dicer as enzyme. Okay. Anyway, let's understand this mechanism of base catalyzed RNA hydrolysis. RNA hydrolysis. Hydrolysis basically means hydro means water, lysis means breaking. Okay, pyrolysis breaking with heat, hydrolysis breaking with water molecules. Okay, so breaking what? RNA with water. So this is called RNA hydrolysis. The enzymes will be known as anyone? Hydrolysis. Okay, these are how they are named. So this they are calling it base catalyzed. Base acid is H plus and base are OH negative. That's why they are using OH negative, and they're calling this as base catalyzed RNA hydrolysis. Okay. So if you add a base to this whole chain of nucleotide you have as RNA, then it will start a reaction immediately. Why? This base will act as nucleophile, having a negative charge. Okay. And I said multiple times a negative charge will try to attack on positive charge. Okay, and what positive charge do you have? You have oxygen as negative and hydrogen as positive. So they will try to remove this hydrogen from hydroxyl group. Okay, and then you will have a negative in oxygen. Okay, they will remove this and in the form of water, and you will have oxygen as negative charge. Now you have a phosphate and oxygen. Okay, this is phosphate and this is oxygen. So of course oxygen has more electronegative than phosphorus, right? In phosphorus and oxygen, oxygen is having more electronegative than phosphorus. Okay, so can you see this? Your oxygen will have a negative charge and this will have a, so this negative will attack to the positive, and this will again break this bond and this will form as negative. Okay, so this is positive, this is negative, and this will make a bond with this oxygen, phosphorus, oxygen. This is oxygen, phosphorus, oxygen. You see that it's a cyclic bond which is formed, and that's why they are called known as mixture of 2 and 3 monophosphate derivatives. Okay, this, and there's a short strain of shortened RNA. Shortened RNA means this bond will cleave and you will have only phosphate from here. So this is called shortened RNA, means they are hydrolyzing, they are making one whole nucleotide shorter. Okay, this is the method to remove one nucleotide at a time. So imagine if you have a polymer of nucleotide and you want to remove this one, you will use a hydroxyl group. It will cleave this bond using this mechanism and remove one nucleotide. Again one nucleotide, one again nucleotide, one nucleotide again, and so on. Okay, so this is called base catalyzed RNA hydrolysis. Okay, we have this thing here as 2,3 epoxy. It's this kind of epoxy group or maybe some other group named so, but the cleavage could be at this point or at this point, at 2 or 3 position. Okay, that's why in your reaction you have a mixture of 2' or 3' monophosphate derivatives. Okay, this phosphate group can either go to second position or third position, and that's why it's a mixture of either 2 or 3 monophosphate derivatives. Okay, cool. Just a mechanism you have to understand how OH will act as nucleophile and will attack to the whole chain of nucleic acids. Okay, cool. Hydrogen bonding interaction. I said you will learn in 5 minutes why there's a complementarity between 5' and 3' and anti-parallel. They are anti-parallel in nature. Okay, so as there's a rule which is A always pairs with T and C always pairs with G, and we call ATCG. Adenine will always pair with thymine and cytosine will always pair with guanine. Why so? We know a purine will always pair with pyrimidine. Okay, so we'll understand why. Because if you have a purine which is adenine, they are two ring structures, and if you have a pyrimidine, it has one ring structure. Okay, similarly, if you have C which is one ring structure, and G which is guanine which has two ring structures. So can you see, if you have a double helix structures over here, if you see there's a double helix, you have 2 and 1 3, 1 2 and 1 3. So the distance between the double helix will be always maintained, because you have a two, you have a one double ring structure and one single ring structure. Similarly, one single ring structure and one double ring structure, and you will always have three rings in your double helical structures, and it will maintain the helical form of the DNA to make them more stable. And that's why purine will always pair with pyrimidines, and pyrimidines will always pair with purines. Okay, let's see how and why. We already knew how. We'll see by hydrogen bonds. Okay, this is one of the most important hydrogen bond in biological system between purines and pyrimidines in the structure of DNA. Okay, it's very easy and it's very difficult. Once you start drawing in the paper, it's difficult. Once you see them in the book or anywhere, it's easy, because you know adenine will always pair with thymine, cytosine will always pair with guanine. But these structures are anti-parallel, and that's why you see sugar in a different form. So if you try to understand, see this has an oxygen, but this has an oxygen. So it's, this is going this way and this is going the other way. So you have to imagine everything opposite. Okay, so let's see how. Let's see the bond between adenine and thymine. Okay, it's very important to understand why the functional groups are present in different positions so that they can make hydrogen bond. Okay, so do you remember this structure adenine? Yes sir, adenine is 6 aminopurine, and thymine is, come on, thymine is, yes, other name, 2,4-dioxy 5-methyl pyrimidines. Okay, can you see that this could be, this is opposite, NCNCCC. You can definitely see everything here. Okay, no matter what. So this is a second position. Will you start always with nitrogen, which is here 1 2 3 4 5, 1 2 3 4 5 6, 5 methyl 2,4-dioxy. It's good, right? Can you see carbon attached with oxygen and nitrogen attached with a hydrogen? They are not attached just for so that you can remember 2-amino 6-oxy. Okay, they are attached so that they can make a hydrogen bond with adenine. Let's understand adenine one more time. We have NCN and CCC and NCN. Okay, this is 7, 8, and 9. This is 6 amino position. So this is 5, this is 4, this is 3, this is 2, this is 1. Even if I don't know, I can try to figure out which are what numbers. Okay, so this is 1 2 3 4 5 6 anti-clockwise, and this is clockwise. Okay, you just have to remember everything in space, because we don't, earlier we tried to like draw on plane of paper, but this is now going three-dimensional. Okay, so this is 6 amino purine. So at 6 amino you have nitrogen attached with 2 hydrogen, and at 1st position you have a nitrogen. So the 6th position amine group will form a hydrogen bond with oxygen of 4th position of carbon, and this first position of nitrogen will form a hydrogen bond with 3rd position of nitrogen. See, and there will be 2 hydrogen bond in between adenine and thymine. Okay, can you see other possibility of forming any hydrogen bond? Can I form a hydrogen bond here? If so, why? If not, why? It could be adenine 3 bond hydrogen. If so, why? And if not, why not? It would be interesting, right? Adenine 3 bond thymine, cytosine 3 bond guanine. Is this hydrogen bond incorrect by some reasons? Yes, no. Because I see a possibility of hydrogen bond for sure. Because the atoms are organized so to form a hydrogen bond, right? Distance is quite very similar to the others too. Not very far not to form a hydrogen bond actually. Answer is very simple. Yes, yes, yes. Tell me. Yes, absolutely correct. So you have to understand at what circumstances you can form a hydrogen bond. Okay. So you cannot form a hydrogen bond until and unless it is attached with a highly electronegative element. It could be either fluorine, it could be either oxygen, and it could be either nitrogen. If you have a carbon and attached to hydrogen, you cannot make a hydrogen bond attached with a carbon. You're getting it? So this bond is incorrect, or you cannot form a bond because this hydrogen is not attached with oxygen, fluorine, or nitrogen. Okay, you are absolutely correct. 10 points, extra credit. So this is incorrect. Why? Because this hydrogen is not connected with a highly electronegative element. Okay, cool. So we can always form two hydrogen bonds between adenine and thymine. Okay, and this attached, this hydrogen bond is formed between 1th and 6th position. Okay, 1 3 and 2 6 4 6. Okay, but anyway, let's see three hydrogen bonds between guanine and cytosine. Do you still remember those structures? Guanine is, yes sir, guanine is 4 amino 2 oxy purines, and cytosines are opposite. Everything is mugging up around. Come on, sir. Okay, okay, okay. If not, let's figure out. Okay. If not, let's figure out, because we already know NCN, CCC, and NCN, right? So this is of course uh, this is 2 oxy 4 amino. This could be 3. This could be 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. 2 oxy 4 amino, 2 oxy 4 amino, or I wrote 4 amino 2 oxy. Remember that? Purine. Okay. And this is other way, which is 2 amino 4 oxy. So this is NC. Always start with N, 1 NCN. Okay. Let's start with NCN, maybe 1, 2, 3, 5, 6. Yeah, because it's opposite. It's opposite direction, right? So 2 amino 6 oxy pyrimidine, right? Can you check if you have the same structures? 2 amino 6 oxypurine, or 2 or 4? 2 amino 4 oxy. Okay, so might you are correct. Let's see this as 1, 2, 3, 4. Okay, now I'm correct. Yes, 2 amino 4 oxy pyrimidines. Okay, cool. Let's see the possibility of hydrogen bond. Okay, you have 2 amine group. This H will pair with oxygen, this hydrogen will pair up with the nitrogen, and this oxygen will pair up again with hydrogen. Any problem? This one, guanine. This one. Oh, it started with carbon, which is incorrect. Okay, okay, okay. Thank you, thank you. I think you're getting it then. So this is NCN 1, 2, 3, 4, 5, 6. So this is 2 amino 6 oxy. Yes or no? 2 amino. Thank you, thank you. 4 oxy purines. Okay. Anyway, but the whole point is you should learn how the structures are made and how they connect with 3 hydrogen bonds. Okay. They are oriented themselves in order. Can you see this hydrogen is attached with nitrogen? That's why they can form hydrogen bond. This itself is a nitrogen, definitely can form a hydrogen bond. This hydrogen attached with nitrogen, that's why they can form a hydrogen bond. This is also attached with nitrogen. If there's a carbon here, you cannot form a hydrogen bond. That's why there are 3 hydrogen bonds between adenine and thymine, and 3 hydrogen bonds between cytosine and guanine. That's why I told it's hard and easy to remember the structures when they are present in anti-parallel order in 3-dimensional order, 3-dimensional space. Okay. Definitely, definitely. Okay, and we'll learn that while calculating the melting point. Okay, that's why if you see a DNA that has more cytosine and guanine and a DNA with less cytosine and guanine, the one having more cytosine and guanine are more stable. Why? Because they have more hydrogen bonds. More hydrogen bonds, more stable. Okay, cool. So let's go ahead and see discovery of DNA structure. I told a slight story of Watson Crick published in 1953, the most famous paper. The structure has novel features which are considerable for biological interest. One of the most important discoveries in biology. Good illustration of science in action. Okay, messy steps of the path to a discovery. Value of knowledge, value of collaboration, value of stealing, value of sharing data, or cost of sharing your data too early. Okay, it's a beautiful story, or you can like see a multiple um, post or vlogs on that. Anyway, the famous Watson and Crick. Okay, or I would recommend to watch a documentary on YouTube. They are they recorded that then when they were very old. It's a beautiful documentary on YouTube. If you see Watson Crick, I might share the link in the blackboard just in case. It's a beautiful documentary. They were sitting in their home and they were trying to explain how they got to a point where they discovered like double helical structure of DNA. And this is a very famous picture. I don't see a picture here, but there's a famous picture of both two standing with a 3-dimensional model of the double helical DNA they made with ball and stick. So the ball and stick model of double helical DNA. So imagine you were figuring out how to orient those nitrogenous bases in space, right? They were doing those for almost 2-3 years by orienting the nitrogen and hydrogen to see if there is a possibility of hydrogen bond formation, so they can stick together. Okay, it's a beautiful story, and if you might find that picture somewhere. This is Rosalind Franklin, more famous. She died at the age of 53 or something by ovarian cancer, and Morris Wilkins. These were the two who provided data for X-ray crystallography. Okay, so yeah. Anyway, and this is the famous picture who lead to discovery of double helical. How to deduce this is not like this as double helical structure. No way. Okay, this is the crystal structure, and it's very hard to uh, get any observation from a single image. And at some point, when they were accused of stealing and scientific, um, misconduct, Francis and Crick defended themselves by saying that I cannot draw I cannot make a 3D model just seeing at one small picture of DNA. Okay, that's also at some extent they already contributed something. It's not easy to just see a picture and draw a double helical DNA with major and minor groups, adenine, thymine. Not only that, the other thing I appreciated about Francis and Crick is they proposed an idea that DNA can replicate. They said a single sentence, there could be a possible mechanism of replicating DNA. They did not say anything about it, but they gave a hint which lead to Griffith experiment and how DNA replication happens. That's a whole other story after 1953. It's biological evolution after discovery of DNA structure. So it's fine. And yeah, this is what happened. This is the different models of DNA, other forms of DNA. It could be A DNA, B DNA, or Z DNA. And these are different forms of DNA with different properties. The most famous and more acceptable was B form of DNA, and this is what we have in our body. B form of DNA. What's the characteristics of B form DNA? This is B form DNA. We have a base pair. How many base pairs? 6 base pair. So imagine if you draw a double helical structure of DNA, this looks like this. You cannot draw a double helical like this, which is very incorrect because it has a major and minor grooves. Minor grooves is this, and major grooves is this. Minor groove is this again, and again it will be a major groove. Okay, and this is the distance between two a pitch of a helix, which is a complete 360 turn at complete 360 turn. A helical sense diameter 26 angstrom is the diameter of the helix. 11 base pair can be present in A form, 10.5 base pair in B form. The distance between two sugars, no, base tilt between normal axis is 6 degree. So like between two base pairs, they have a tilt, and they all tilt to form a helical structure. These are the property you have to remember. 2 prime endo and 3 prime endo. Okay, these are different forms of sugar they have. This is called sugar pucker conformation. I already made a video about this, but anyway, this is anti conformations of glycosidic bond. These are just the information you have to remember. Z form of DNA, it's called, they have 4 different nitrogenous bases attached to them. That's a whole other story, but yeah, these are different forms of DNA. Not much to remember, but you should remember that the DNA which is more common is B form. Okay, two chains are different in sequence, 5 prime to 3 prime. As I said, one runs 5 prime to 3 prime, other runs 5 prime to 3 prime, but they are anti-parallel. Okay, so this is at cytosine and guanine with 3 hydrogen bonds, and adenine and thymine with 2 hydrogen bonds. Okay, complementary and anti-parallel. Okay, complementary means they complement each other. Okay, and anti-parallel means they are not running in the same direction. Okay, replication of genetic code is just one sentence given by Francis and Crick model. Okay, they said there could be a possible mechanism to replicate the DNA, and yeah, and DNA replication needs an enzyme which is called DNA polymerases. Ases means enzymes again. Strand separation occurs first. This is the whole story of how they made a fork and then they replicate DNA. One is small pieces, one is continuous, and so on. There's a whole other story of molecular biology, how we can learn replication, transcription, and translation. A new era of molecular biology, cellular and molecular biology. You don't need to learn that in biochemistry class anyway. But this is just wanted to know how DNA replication happens. The fork opens and they form a parent strand which is already there, a parent strand already there, and they form a new strand in opposite direction. Okay, and this is called as replication fork. Okay, this is not actually what how DNA replication happens. It's a different, this is an enzyme which is involved, which is called DNA polymerases, and it's a whole other story. Okay, but yeah, messenger RNA, which is called mRNA. The other two RNAs are tRNA and rRNAs. Okay, they contains uracil in place of thymine. So that's the other difference between RNA. RNA has uracil, whereas DNA has thymine. Okay, that's the other difference between DNA and RNA. One first difference was deoxyribose and ribose sugar, and this is the other difference, having a uracil in place of thymine. Okay, tRNA are helpful in transferring of genetic information. They have amino acid in their top, and you will learn that in process of translation. Okay, if you see how DNA replicates. Palindromic sequence. Okay, palindromic sequence are sequences that can be similar when you read it from the different directions. Okay, similarly, if you say papa, no, papa is not a word, but any word which is palindrome to you. Hmm? EYE, very good. EYE is a palindrome word, very nice. Any other? DAD, dad is also a palindromic word. Mom is also a palindromic word. Okay, SOS is also palindromic word. Pop is also palindromic word. So these are the palindromic words. But in DNA, if you have a sequence of GAATTC and GAATTC, this is 5 prime to 3 prime, and this is 5 prime to 3 prime, but they are same when you read them from different directions. So these are called palindromic sequences, okay, because they can be read same if they are in opposite direction, okay, and they are complementary to each other. See, okay. Another palindrome, TTA GCAC, TTA GCAC. See, it's a palindrome, TTAG CAC, and then TTAG CAC. Okay, it could be a palindrome or it could be a mirror repeat. Mirror repeat means if you go from here to here, it's same, here to here, it's same. So they are different. Palindrome is this opposite sides, and this is mirror repeats, which is same at the same strand. They have a mirror image of each other. If you place a mirror here, it's the same thing. Okay, it could make a hairpin structure, because you already know that strands of DNA are complementary to each other. So if you have a long strand, they can make a coil by themselves and make a folded structure. Here adenine and A and C cannot pair to each other. That's why they are making head of hairpin structure. Okay, yeah, they will make a bulb because they are not complementary to each other. Okay, they cannot make a hydrogen bond. Okay, and this is the neck of hairpin, and this is base of hairpin. So this is hairpin structure of RNA. Why? Because they are single stranded. Okay, so these are some more of the complex molecules of DNA or RNA. Can you see that? So imagine the small base pairs with those structures they have. They are present in a very complex form. So it could be a quadruplex model or triplex model or anything, but yeah, these are the complex structures of DNA or RNA. You see that this is all nucleotides clumped together. Okay, similarly this and similarly this. This is guanine, but there could be modifications of guanine. As I said, there's a there is a cysteine, but there could be a selenocysteine, and there could be something else with a modified form. This is 7-methyl guanine. So at the 7th position, they have a methylation. See, similarly, N dimethyl guanine. So at this position, they have 2 methyl groups. Okay, adenine. So there could be some modified nitrogenous bases that could be present in DNA or RNA. Okay, just the modified forms of amino acids. DNA denaturation, covalent bonds, hydrogen bond, and base stacking. These are the 3 steps. DNA denaturation. What do you mean by DNA denaturation? If you have 2 strands of DNA, and if you provide certain temperature, it will unfold and make 2 strands of DNA. This process is called DNA denaturation. It says covalent bonds remains intact. It means the phosphodiester linkage they have is not broken. Okay, the only thing which is broken is hydrogen bond. Okay, so this is called hydrogen bonds are broken. Yes, that's why two strands separate. Base stacking is lost because of course the helical structure is no more. Okay, DNA denaturation can be induced by high temperature or change in pH. Okay, it's reversible and the reverse process is called annealing, which is making one double helical structure with two separate strands of DNA. Okay, see this process, this is called denaturation, but the reverse is annealing. Okay, and this is complete separation of two strands of DNA, separated DNA in the form of random coils. Okay, they are very unstable, they always need their other half to be in the form of double helical structure of DNA. So imagine if you heat the structure of DNA molecule, it will separate into two pieces, but once you remove the extra heat, it will again try to attach by themselves. You don't need anything else, and then they will form a double helical structure of DNA. This is nature of, this is beauty of nature, how they can regain their shape once they are affected by the external forces or heat or temperature or pH. Okay, so yeah, DNA denaturation, thermal denaturation means giving heat. Okay, and the temperature is called Tm, which is melting temperature. So polymerase chain reaction is one of the common example how we denature the DNA and replicate DNA in different forms. So imagine if you have one copy of DNA and you have done the denaturation, separated two strands and provided all the materials here with the enzymes called DNA polymerase, all the nucleotides, nucleosides, phosphate, energy and everything. This can form their other part, other half. Yes, this can form their other half like this. See, and this can also form their other half, right? And now you will have two copies of DNA from one copy of DNA. Now again you will repeat the same process. Now you have two strands of DNA and you will go to 4. Now you will again repeat the process, you have 4 strands and you will get 8. Now you repeat the process, you will get 16. So it replicates in the 2 to the power of n. Okay, so if you run 4 cycles of DNA, it will be 2 multiplies 2 multiplies 2 multiplies 2, 2 to 4 to 8, 16, 16 copies of DNA. And this is how we detect nucleic acids presence in any sample. So imagine when we were facing COVID, okay, it was a pandemic, and we are trying to understand the structure of RNA viruses they have, right? If I have one copies of RNA, we will run a cycle of PCR, which is polymerase chain reaction, and we get 1,000 copies of the same RNA, and we can easily detect those copies because we already know the structures of those RNA. And this is how sampling and detection of those nucleic acid sequences can happen using PCR, which is polymerase chain reaction. Okay, so yeah, this is a melting temperature. It could vary in different samples. So which one has a higher melting point, this one or this one? This sample, this is 1 and this is 2. Which one is having higher melting temperature? 2, because this is 85 and this is somewhere between 85 to 87, where 50% of DNA is denatured. Okay, this could be 83.5 and this is 85. Okay, so which one is more resistant for denaturation? 2 is more resistant to denature because more temperature is required to remove two strands separate. Okay, so this is how Tm is observed. If you have high Cg content, means you need more energy to break more hydrogen bonds, and that's why they have higher melting point. Okay, makes sense? So imagine if I give you a polypeptide sequence with 20 Cg bonds and one polypeptide with 10 Cg bonds. Which one is more stable? Yeah, 20, because it has more Cg bonds. Okay, pH, of course, ionic strength. Salt increases the melting point. Why? Because if you have a salt, they make stable electrostatic interactions between them and it makes them stable, and that's why you need more energy to break them apart. Okay, makes sense. These are few basic. This is GC correlation with melting temperature. If you have more GC content, your melting point will go up. It's a linear increasing relation. Okay, makes sense. This is how a loop is formed while replication of DNA. Okay, AT rich region melts slower, is lower at lower temperature than GC rich regions, which is because AT has two bonds and CZ has. So if you start heating a DNA, which region will break first? Which has more AT content. And this is, I think this is replication of E. coli bacteria. So E. coli has a DNA which is present like this, and you see all these loops. See, these are open structure where replication is already happening. Okay, DNA replication is a whole different story. It's a complete new world. Okay, if you want to know more, just email me. I can teach you more about DNA replication, but anyway, this is not the topic in biochemistry. Two near complementary DNA structure can hybridize for sure, making a double helical complex. Okay, amplification of DNA is making more numbers of DNA. Detection of specific DNA molecules by radioactive detection of fluorescent DNA chips. They are called nano arrays. Okay, they are new techniques for detection of nucleic acids, mostly for diseases. Okay, if you have a viral infection and you want to know which virus affected you, these techniques can help you identify those viruses. If you have an infection of human papilloma virus for cervical cancer, you can definitely know that using these techniques. Okay, if you have a HIV infection, you could definitely track those using these techniques. Okay, I hope not anyone. And antisense therapy, you can find a evolutionary relation between two species, how they are related to each other by looking at their DNA and the similarity between them. And yeah, it's a useful technique. If you mix two sample 1 and 2, and if they are complementary to each other, they will make a hybrid complex between 1 and 1 itself, 2 and 2 itself, and 1 and 2 itself. So you will see three types of DNA: one complete red, one complete blue, and blue and red. See, so these conclude that the structure of DNA is complementary to each other. They do not require their own species to hybrid, and that's why viruses intercalate with our DNA to produce their protein. So they utilize all whole machinery to make their own food and protein and their population. Okay, so this is what a code is universal. What I mean, genetic code is universal. Deamination, depurination are two different methods. Deamination is removal of amine group. Depurination is removal of purine groups or purine nitrogenous bases. Okay, see here what I mean by deamination is removal of this group. Can you see that? Deamination, same thing. Deamination, same thing. Okay, deamination, same thing. So conversion of NH2 into double bond O is deamination. Amination is removal. D means removal, amination means removal of amine group. Simply depurination, which means removal of purine. So imagine if you have this is purine, this is sugar, and this is phosphate, and you remove this thing, you will have this sugar with phosphate and purine, and this process is called depurination. Simple, just the names. Okay, molecular mechanism of oxidative and chemical mutagenesis is how we use different oxidative agents to damage the DNA, chemical alkylation, or this is called defense mechanism they have to protect the DNA, which is uh, which can protect the all the alteration that happens. Okay, I don't know whether you have gone through this or not. Alkylating agents, these are called mutagens. Okay, anything that can cause mutations. So whenever you eat or drink or use anything, look at the labels, what chemicals they have. Your dyes, the colorful drinks you take can have yellow 6, yellow 5, red 9. Those codes are cancerous codes. Okay, they could be mutagens that can alter your DNA, change your DNA to something else. Maybe some process is affected. You will not know that until that affects your body in some other way. Until you get the symptoms, it's already too late. Okay, so I'm not scaring you, but that's the reality, that you should always check labels, what you eat and what you drink. Colorful things are dangerous in general, universal statement. Colorful snakes are dangerous, colorful frogs are dangerous, poisonous, colorful drinks are dangerous because they might contain mutagens. Those are all artificial chemicals we synthesized as humans. Nature has never made those things. Okay, nature had made colorful things, but not colorful chemicals we use in our daily life. Okay, so just be careful, do not use any mutagens. Although body has defense mechanism to protect all those mutations, that's why we are alive. Otherwise we would have eradicated from earth using those things. Okay, so UV radiations can also damage. Ionizing the radiations, for sure. If you are using X-rays and gamma rays in your lab or anywhere, be careful, try not to be exposed to them. Also, UV radiations are known to cause cancer. That's why we use sunscreen all the time. When you go to the sun, you use sunscreen to make sure you are protected from UV radiations from the sun, because they can cause skin cancer by damaging your DNA. And next time when people ask you why do you use sunscreen, your answer could be, it induces dimerization in my pyrimidines. That's why I use sunscreen. Can you say that? So whenever someone asks you why do you use sunscreen, you will say that it caused dimerization in my pyrimidines. And what are the pyrimidines? Which is C, U, and T. So dimerization means C can form bonds with C, and U can forms with bond U, and T can form bond with T. So imagine what you are protecting yourself from dimerization of your pyrimidines. Okay, so next time you use sunscreen, just remember, if you don't use your sunscreen, your UV rays you can dimerize your pyrimidines, and it will be a mutation. Okay, makes sense? At least you learn one thing today, which is, why do we use sunscreen? Exactly, to protect dimerization of your pyrimidines. Okay, ionizing radiations can cause ring opening, which is opening of the ring of one of the nitrogenous bases or sugar or whatever. And cells have, of course, mechanism to protect you. That's why if those accumulations are accumulated, it will be aging or carcinogenesis. You might look old, or you might get a cancer. So you protect your pyrimidines. Okay, this is a kink, which is, see, these are dimerization of T and T. Okay, why? Because this person did not use sunscreen. Okay, so anyway, I cannot be more funny than this because I'm a very serious guy. Okay, so yeah, I think I already have gone through this. What is a nucleotide monophosphate? If you have just one phosphate, monophosphate, two phosphate, diphosphate, three phosphate, triphosphate. So if I say NMP, nucleotide monophosphate, and DP, nucleotide diphosphate, and TP means triphosphate. And I think you know this person very well. ATP, yes sir, adenosine triphosphate. Now you get that why they are called adenosine triphosphate, because they have adenosine in them and they have three phosphate groups. Okay, similarly, if you have ATP, you have GTP, CTP, and UTP. The only difference is their nitrogenous bases. Okay, if you remove one phosphate, it will be diphosphate, one more phosphate, monophosphate. And this is alpha, beta, and gamma. Do you remember alpha amino acids, beta amino acids, and gamma amino acids? They are very similar to that. Okay. Other functions can be coenzymes. Coenzymes, they can form coenzymes A, which is pantothenic acid. Anyone who knows what is a pantothenic acid? Another reasons why you should take multivitamins. You see where they are making a role, or why they are micronutrients and why they are useful. If you do not take your micronutrients or multivitamins, these multivitamins act as cofactor of the enzymes. So you do not do everything just with your enzymes, you need those cofactors to act efficiently. Okay, and these are where vitamins plays a role to be a cofactor in all the enzymes activity. Okay, anyway, riboflavin, you know what is riboflavin? Vitamin B, B1 is riboflavin, I'm not sure, but I have to check. I used to remember all the names of um, B complexes: riboflavin, pantothenic acid, cyanocobalamin is B12, folic acid is B9, B9 is folic acid, B12 is cyanocobalamin, which contains cobalt. Okay, B5 is pantothenic acid, and B1 is thymine, B2 is riboflavin. I remember this from my memory. B3 is something else, I don't remember now. Niacin, B3 is niacin. B4 is sorry, I cannot remember all of them now. So, but yeah, you can remember the names of those amino acid B complexes for sure. So these are all other components of nucleic acids which are functional as enzymes and coenzymes and so on. Okay, not very important, but cyclic AMP act as secondary messenger. We discussed about this cyclic GMP or PPCGP, PPGP. This is very important. I was in a seminar who worked on PGPP last week, but this is guanosine, sugar, and phosphate, 2-phosphate, right? That's why they are diphosphate. At least you can compare and understand what is what. Okay, summary: we understood what is the function and of nucleotides and nucleic acids. Of course, we know the names or structures of common nucleotides. We understand structural basis of DNA function, denaturation of nucleic acid, and chemical basis of mutagenesis. Okay, any questions? Preguntas? This is only thing I know in Spanish. No? Yes? If not, then thank you so much, and all the best for today, or maybe on Sunday you have a test.